Yeah.
Go ahead,
Okay,
Vesicles.
our next speaker is a poster or speaker, Genevieve Anghileri, I think it's right or not. Sorry, from Loughborough University. So 7% and then questions.
Just to double check that everybody can see my screen okay. Yeah. Okay, so I'll begin. Thank you, first of all, everyone, for being here for my talk today. As they've said, my name is Genevieve Anghileri, and I'm currently supervised by Dr. Owen Davies at Loughborough University. My project is titled Determining the Function of Matrix-Bound and Secreted Vesicles in Bone Mineralisation. Just before I start, I'd like to thank the Veda Lab in the UMC Utrecht for recently hosting me on a short placement funded by UKEV. Some of the data I will share today was collected during that placement. So I guess we start with, why would you want to study EVs in the context of bone mineralization? So bone actually is the second most commonly transplanted tissue worldwide. So you might be surprised to know that actually a lot of the mechanisms underpinning bone formation are still quite poorly understood. And this means that in instances where bone mineralization is hampered, such as osteogenesis, osteoporosis, and large injuries, therapies are still quite limited. So bone grafts have traditionally been used, both biological and synthetic, with recent inclusions of factors such as BMP2 in order to enhance the mineralization. But with significant side effects and limitations, researchers have more recently looked towards stem cell-based therapies, of which we know the mode of action is most likely EVs. So yeah, this leads me on to say that the matrix-bound vesicles located in the early osteoid are actually one of the first EV populations to be discovered back in 1967 by two independent researchers that published it in the same year. So these matrix-bound vesicles are believed to be released from bone-forming cells. They're then believed to embed in the early osteoid, and actually, as you can see in the diagram, form the environmental niche for the early mineral formation. Once this mineral has matured, the matrix-bound vesicle, or MBV, is no longer required, so it's thought to burst, and the mineral is then laid on the extracellular matrix. Though these MBVs seem to be so important, they're actually largely undercharacterized in terms of origin, content, and pro-mineralizing function. And specifically, they've not really yet been compared to the more commonly cited exosome-like secreted vesicles that many of you will routinely study. We're calling these SEVs. During my PhD, I aim to explore the biogenesis, content, and specific pro-mineralizing function of the MBVs and the SEVs, eventually with the ultimate aim of defining an optimal EV population for osteoregenerative therapeutics. So I started by mineralizing cultures of both MC3T3 preosteoblast cells and human MSC stem cells differentiated down an osteoblast lineage. Most of my early optimization work was done in the MC3T3, but following confirmation that both cell lines were fairly comparative in terms of results, most of the data I'll present today is from the MSC cultures. So as you can see in the alizarin red stain, this confirms the mineralization of the cultures, and also the alkaline phosphatase ALP, known to be an early mineralization marker, increases over the time course of the cellular mineralization in the cell lysates, as you would expect. So we isolated matrix-bound vesicles, MBVs, and the secreted SEVs at various time points across cellular mineralization, from the extracellular matrix and the conditioned medium, respectively, via ultracentrifugation. Matrix-bound vesicles were also isolated via immunomagnetic isolation to be positive for annexin-5 and alkaline phosphatase, as we'll discuss later on. As you can see on the right-hand side, nanoparticle tracking analysis shows not too dissimilar size profiles between the MBVs across the top and the SEVs along the bottom, with maybe the only difference being a slightly broader profile in the MBV populations. So now we move on to more of the EV analysis. So an assay revealed a markedly increased content of alkaline phosphatase in the MBV samples compared to the SEV, probably pointing towards an enhanced promineralizing capacity in these MBV samples. We confirmed underneath via super resolution microscopy that the alkaline phosphatase that you can see in purple co-localizes with the lipid dimemglow, which you can see in blue, showing that the alkaline phosphatase we're measuring is in fact EV bound and not free floating. We then confirmed again via super resolution microscopy that these ALP positive MBVs and SEVs also seem to co-express annexin-2 with a markedly enhanced or significantly enhanced percentage of co-expression of annexin-2 with ALP in the MBVs compared to the SEVs, again, possibly pointing towards this enhanced pro-mineralizing capacity, as we know that annexins are calcium channels and therefore likely important for this mineralization function. We then confirmed via immunomagnetic isolation followed by Western blots that annexin-5 positive MBVs are also expressing ALP, and ALP positive MBVs are also expressing annexin-5. Once we showed this enhanced mineralization capacity via protein, we also went on to show it via transmission electron microscopy, which you can see in the bottom. So in the later time points of cellular mineralization, the MBVs appear to demonstrate enhanced electron density. We don't yet know that this is mineral. We haven't done any physical characterization, but it's definitely a good sign that these MBVs have an enhanced mineralization capacity. Finally, we went on to look at the biogenesis of the MBVs and the SEVs. To start with, again, via immunomagnetic isolation of the MBVs, followed by Western blot, as you can see on the left, the annexin-5 positive MBVs and the alkaline phosphatase positive MBVs also express CD9. Again, we went on to confirm this via super resolution microscopy, and we showed that the ALP positive MBVs and SEVs also co-express CD9, CD63, or CD81, again, as you can see on the right-hand side in the images. And again, the percentage of co-expression was significantly higher in the MBVs compared to the SEVs. We also have early Western blot data that seems to demonstrate presence of both ALEX and TSG101 in both the MBV samples and the SEV. So this is early encouragement that despite popular opinion in the field, the matrix-bound vesicles with this pro-mineralizing function that we are so far demonstrating may not be formed in a too dissimilar way to the more exosome-like SEV samples. This is very much a work in progress, so I do welcome comments, suggestions, and questions, but I do hope I've at least sparked some interest in the matrix-bound vesicles. Thank you.
Thank you for your presentation. Any questions for the audience?
Yeah, I have a question. Brilliant work. I mean, this is I wasn't on yesterday for any of the sessions, but today I think this is the first time I've seen some super resolution microscopy. And bravo, those images look great. Do you want to just explain really quickly what's so unique about those images?
Yeah, so these images are obviously at a single vesicle level, which is quite difficult to get, especially with the fluorescence. Obviously, you can do it partially with the TEM, but here we can really see the co-expression of the two fluorophores at the single vesicle level. And because they blink, it does give you quite a good idea of size and shape. So you can be fairly confident that what you're seeing is truly an EV.
Quick question.
Thanks.
And I don't know if it makes sense, but since you are working with cells with mineralization, and do you need to have any kind of different approach, like to isolate EVs, something different from like normal cells that do not create mineral?
Yeah, so obviously for the secreted exosome-like SEVs, we just isolate them as normal from the conditioned medium via ultracentrifugation. But for the matrix-bound vesicles, because we need to release them from the extracellular matrix, we just do a simple collagenase digestion. And then, using the differential ultracentrifugation, we can separate out the matrix-bound vesicle part from the remaining debris.
Yeah, that was it. Thank you.
Yeah.
Yeah, we have a question of Nishta. Maybe you can ask directly.
Okay.
Thank you.
Nice work, Genevieve. Maybe I missed it in your talk. How are the MVVs and SEVs isolated? I couldn't quite catch that.
Yeah, so we isolate, in most of the work, I've isolated them both by ultracentrifugation. Again, like I said, the SEVs come from the conditioned medium, and the MBVs, we have to release them from the extracellular matrix via a collagenase digestion. So yeah, mostly differential ultracentrifugation with the immunomagnetically isolated MBVs, obviously being isolated immunomagnetically with dynabeads.
Okay, thank you.
Yeah.
Okay, thank you. Thank you for your talk. Samek will present the next speaker.
Yeah, absolutely. So for the next speaker, we have Victoria Carololi from the Instituto Multidisciplinario de Investigaciones in